Okay, thank you, Megan. Really a pleasure to be here to speak to you all about the genetic basis of heart attack. Let me jump right in. You can leave time for questions at the end. So before I start, I just want to just acknowledge different individuals over the years who's contributed to the work. I'm going to be talking, summarizing more than a decade of work. And on the left is my own laboratory. This is from a couple of years ago, unfortunately, but individuals over the years have contributed heavily to all the work. And then on the right, of course, is the Broad Genomics platform. Almost all of the data that I'm going to show you was really generated through the platform. And I've had a very productive and wonderful relationship with Stacy for now more than 15 years, and Namrata for almost a decade as well. And she's really helped lead most of the projects that I'm going to show you. And then we've had a host of collaborators over the years, both for the heart attack studies as well as the lipid studies that I'm going to show you from all over the world. So thanks to all of them. Okay, with my history background, I'm going to start with this slide. Two men who suffer from cardiovascular disease. On the left is Jim Fix. On the right, of course, is Churchill. So Fix is credited with starting America's fitness revolution. In the late 70s, he had a very famous book called The Complete Book of Running. Unfortunately, he died while jogging in Vermont at the age of 52, and he died of a heart attack. Of course, Churchill is kind of a poster child for bad health behaviors. He was always had a cigar in his mouth. He drank a lot. He's obese, but he lived to the ripe old age of 90. And there's actually a funny story about Churchill. He was around World War II, was visiting the king of Saudi Arabia, and was told that in the king's presence, because of the Islamic faith, that Churchill could not drink or smoke. And so he famously said, according to his religion, it was a sacred right to smoke and drink before, after, and during all meals, and in the intervals in between them. So he actually was allowed to smoke and drink, I think, at that dinner. But anyway, he lived to a very old age despite all of these risk factors for heart disease. And so these two men, I think, actually highlight two questions that we think about for we all work on and kind of think about almost daily, is that why are some people at risk? So why do some get disease at an early age? And then conversely, why do some seem to be protected despite exposure to risk factors? And so are there pathways, for example, are there mutations that might confer resistance to disease? So these are the two questions I'm going to address in the context of cardiovascular disease. So can we identify those at high genetic risk and offer preventive interventions? And then can we find pathways that actually protect against disease and potentially develop medicines that mimic them? Okay, let's start with the risk part. And then to get everybody up to speed on the disease process, so this is the problem we're going to be talking about is coronary artery disease and myocardial infarction. It's the leading cause of death worldwide. It is a heritable, common, complex disease, and it has two phases. So shown here is the heart, and on the surface of the heart run the arteries that supply blood to the heart, the coronary arteries, and here's one of those arteries in cross-section. And the wall of the artery is where the problem is. So here, over time, during the chronic phase of the disease, there can be a buildup of plaque in the wall of the artery. And then there's an acute phase where the lining, the interior lining of the blood vessel can burst, exposing the subendothelial collagen to the flowing blood, and that's a very potent trigger for blood clotting, for thrombosis. And if that blood clots, that blood clot stays there for more than 20 minutes or so, then the heart muscle that's served by that artery can die. That death of heart tissue is called myocardial infarction or heart attack. And that death of heart tissue can be detected by symptoms, EKG change, characteristic changes on this surface electrocardiogram, and then elevation in blood protein levels. So two parts to the disease: a chronic phase over decades, development of atherosclerotic plaque, and then an acute phase of plaque rupture, in situ thrombosis, and then necrosis of the myocardium served by that artery. All right, and then in terms of additional background, there are some risk factors that have been studied for a while that put people at risk for the two phases, particularly the chronic phase, the buildup of plaque in the artery wall. And these are two of the risk factors. Shown here on the bottom is cholesterol, shown here on the top is triacylglycerol. And these are substances that are essential to cellular function. These substances are insoluble in water. These are lipids, insoluble in water. So the body needs a transport system to actually carry these water-insoluble substances in the bloodstream. And the transport system are called lipoproteins. And so they're schematized here, a phospholipid monolayer. In the center is a neutral lipid, either in, orange cholesterol or blue triglyceride. And there's a protein embedded in the coat of the phospholipid monolayer, either ApoA1 on this type of lipoprotein, and then ApoB on all these other lipoproteins. The lipoproteins are named based on either the density or the lipid it carries in the middle. HDL is high density lipoprotein. It's small, it's very dense. There's low density lipoprotein, LDL. And then these lipoproteins are named based on the fact that they mostly carry triglycerides. They're called triglyceride-rich lipoproteins. And then lipoprotein little a is kind of a cousin of LDL, which is basically an LDL particle with this additional protein, ApoA, added on to the ApoB. Okay, with that as background, let's move to considering the genetic basis of heart attack. So if you think about heart disease and you think about myocardial infarction, the average age of a first heart attack in the United States for a man is 65, for a woman is 72. But some individuals get disease at a very early age. So for example, like Fix, who had a problem at age 52. So if we just set the cutoff, let's say roughly 55 as early onset disease, this is the group that we focused on over the last decade or so. The reason we focused on early onset heart attack is that it's much more heritable. There's a larger genetic component, and the disease later in life has much more of an environmental component. So to understand the inherited basis, we focused on early onset disease. So if you think about why somebody might develop heart attack at a young age, and what are the genetic paths to early MI, there are three major reasons. One is a monogenic model, where a single mutation is sufficient to lead to early MI. Another is a so-called polygenic model, where the additive effect of many variants can lead to early MI. And then the third model is a somatic mutation. So these are mutations not that you inherit from your parents, but actually that are acquired during your own life in specific cells. And so I'm going to walk you through the contribution of each of these models to early MI. So let's start with the monogenic model. So here's the experiment that we've done to address that. We took about 5,000 individuals with early MI, 5,000 control individuals without heart attack, and performed exome sequencing across all 10,000 individuals, and then systematically searched for risk mutation signal in each gene across all these individuals. So the analysis here is relatively straightforward. We're comparing the frequency of mutations in gene, let's say gene 1, in cases compared to controls, with the expectation that if it's a risk gene, there would be an aggregate more mutations in that gene in cases compared to controls. And we do that for gene 1 all the way to gene 18,000. And then for each gene, we can plot basically the statistical evidence that there's a deviation in frequency between cases and controls, deviation beyond chance. That kind of plot is shown here. On the x-axis is the expected p-values for each of the genes. On the y-axis is the observed p-values. Each dot represents a statistical signal for a gene in terms of a comparison of mutation frequency in cases compared to controls. And then the red line is the null distribution, the unity line. And you can see most genes basically fall along the null distribution. So that means they're not involved in heart attack risk. But some genes stand out above the expected p-value distribution. And one gene here is the strongest in the genome in terms of risk for MI. And that gene is low-density lipoprotein receptors. So this is a gene that is critical in terms of regulation of the level of LDL cholesterol in the blood. So to quantify that, about 1 in 200 patients in the world, people in the world, basically carry a mutation in this gene. And as a result of carrying this mutation, they have about 3 to 4-fold increased risk for heart attack. In addition to LDL receptor, there are several other genes that we've identified over the years that affect MI risk using this monogenic model. Another gene is called lipoprotein lipase, and this gene present in 1 in 5,000, mutations present in 1 in 5,000 individuals, and these individuals have higher level of blood triglycerides and higher risk of MI, this degree. Similarly, apolipoprotein A5 is another gene, 1 in 3,000, 4.5-fold increased risk. And lastly, this lipoprotein a that I mentioned earlier, about 1 in 100 have heterozygous deficiency, and then about a 3-fold increased risk if you carry a mutation. So in aggregate, across these 4 genes, about 2 to 3% of all early MI patients, you can identify a mutation in one of these 4 genes, and representing the monogenic model. Now, so that's really where we're at in terms of thinking about strong effect alleles and risk of MI. What you can see now then is if you have 5,000 individuals and you're able to explain, let's say, 2% of them based on a monogenic model, that leaves a lot that's unexplained. Like why did all those other individuals have an MI at a young age? So one possibility is the polygenic model, and here is the experiment that we've done over the years, several of these. This is the latest summarizing all of the data. And here we looked at about 60,000 cases and about 120,000 controls, and performed genotyping rather than sequencing, and compared systematically the allele frequency difference between cases and controls across all these polymorphisms, these common polymorphisms, and millions of them across the genome. And the results of these kind of studies are, as you've probably seen before, are represented on these Manhattan plots. On the x-axis of chromosomes, and the y-axis strength of statistical significance. And the line here represents the genome-wide threshold. Each dot here, rather than representing a gene in the last analysis, here each dot represents a specific site in the genome, a polymorphism. And you can see that there's a bunch of dots above the genome-wide statistical threshold. There are about 95 different gene regions that are pointed to by these variants, these DNA sequence variants. And so there are now 95 loci for heart attack, gene regions for heart attack. We can now think about what are the biologic pathways that are pointed to by these 95 gene regions, 95 variants, and you can group them according to the biomarkers or risk factors or processes that these genes, these variants relate to. And almost 15 out of the 95 relate to either LDL cholesterol and lipoprotein or the triglyceride-rich lipoprotein. So we have an unbiased survey of the genome for common variants related to disease. You find 95 things, and then 15 of them relate to either LDL and also relate to LDL and triglycerides. Here are some other pathways that you can sort out. But then there's a lot, probably more than 50% of the gene regions of the variants don't relate to known risk factors, suggesting there's a lot of potential new biology that we can learn if we could better understand what these SNPs are pointing to, what these polymorphisms are pointing to, what mechanisms. So that's work that's ongoing in the lab in terms of identifying and prioritizing these loci, and then digging down in terms of functional biology. And then there's a part of the lab that's also focused on potentially medicine development based on these new non-lipid loci. But that's a lot of work. That work is ongoing, and it takes a bit of time to complete. But ahead of actually knowing the biologic pathways, one potential utility of knowing the risk variants is actually using the variants to identify those who might be at risk for early MI. And so risk prediction using the polymorphisms as features. So that's a concept that we've kind of looked at for a number of years now. And each variant is actually a pretty small effect. So if you take a look at these variants at this locus that is the strongest, quote unquote, in the genome for MI risk in terms of common variants, the effect size of that variant is only a 30% increase in risk. In contrast to the Mendelian mutations I mentioned earlier, they're about 400% increase in risk, a fourfold increase in risk. But the difference here is that there are 95 of these variants, there's not just one. So you could imagine potentially summing up all these variants into a score, and the idea is a polygenic risk score. If you have n polymorphisms associated with disease, and each person carries 0, 1, or 2 copies of the risk allele at that site, then a score ranging from 0 to 2n can be calculated for each person. And you can weight that score based on the effect size of each variant. And we and others have done this over the years. This is initially 9 variants, this is 12 variants, and here there's 55 as the number of variants have expanded. And then most recently, we've really latched on to a concept that the heritability of the disease lies not just in the top of the list, so those variants exceeding this genome-wide threshold, but there's a lot of signal actually in all of the polymorphisms below the genome-wide threshold as well. In fact, if you take all the variants, all the common variants, you can explain a lot more of the heritability than just looking at the top of the list. So that suggests that we may be able to improve our predictive ability by expanding the feature set for prediction from just 95 to the full set of polymorphisms. And that's what we've done now. Now we've expanded to a genome-wide set of almost 6.6 million variants for risk prediction. And that's work led by Amit Kher in the lab up on bioRxiv as of late last year, and I'm going to show you that in the next few slides. So the hypothesis for this work is that a polygenic score, including a genome-wide set of SNPs, can identify individuals at risk and possibly at risk equivalent to the effect size of a monogenic mutation, a single mutation. And so the starting point for this work is first a training data set where we know the effect size for each of millions of common polymorphisms. So that's 6.6 million variants from that genome-wide association study that I just mentioned that was published. 60,000 cases, 120,000 controls. Okay, so we have a list of SNPs, we have a list of effect size for all the variants. Second step is to construct scores based on those variants and test those in a validation data set. And we turn to this UK Biobank study, which is about 500,000 people, where we know the clinical information, we have the genetic data, and of the first part of UK Biobank, about 125,000 people, 4,000 of them had heart attack and 120,000 didn't. We used this data set to validate the scores. We tested 20 plus scores in each one of these, in this data set, and found the best one in terms of predictive ability. Then we took that score and moved it to a testing data set of the remainder of UK Biobank, a separate set of individuals of about 300,000 people. And here, 9,000 had heart attack and 288,000 didn't. And we were able to actually have almost exactly the same predictive power in this new independent data set, this testing data set, compared to the validation data set, which is kind of what you're looking for in prediction. Basically, you develop a score, you validate it, and then you move it to another sample to say, hey, does the predictor hold in this new independent sample? And it did here. I'm going to show you basically the data from this testing data set. So in each person, each of about 300,000 people, we've calculated a score. Okay? We can plot that score as a distribution in the population across all 300,000 individuals, and here's what the score looks like. Okay, it's a beautiful bell curve, like many other traits in the human population. So if you plotted height for 300,000 people, this is what you'd get. If you plotted LDL cholesterol for 300,000 people, this is what you'd get. So here, instead of plotting height or LDL cholesterol, we're plotting one's quantitative liability to heart attack based on all these polymorphisms. Okay? And seeing the score, you can now ask a couple of questions. How does this score compare or relate to the known risk factors we use in clinical practice to measure risk? So that's like LDL cholesterol, blood pressure, diabetes status, and so forth. These are the things that your doctor looks at to understand risk for heart attack. So here's the correlation coefficient between the polygenic score on the x-axis And on the y-axis is something called an ACC-AHA pooled cohorts equation. So this is a risk equation that doctors use in the clinic to basically measure risk, and this includes blood pressure, LDL cholesterol, and so forth. What you can see is for a set of individuals, there's almost no correlation between the genetic liability score and the existing risk factors that we use in clinical practice. So this means that the genetic score is actually capturing something quite orthogonal to the things that we use in everyday clinical practice. Now, the next question that comes up is, using this polygenic model, can we actually identify a subgroup of individuals that are at high risk, and particularly at risk equivalent to the monogenic mutation, which was, as I said, fourfold for 1 in 200 in the population. And so a couple of different ways I'm going to show you this data. One is here is percentile bins. So on the x-axis is a percentile of polygenic score. So this is the lowest group of polygenic scores, and this is the highest group. Each dot here represents 1% of the sample. So 1% of 300,000, it's about 3,000 people are in each bin. And on the y-axis is the proportion of people in that bin that have heart attack, have had heart attack in the UK Biobank. What you can see is that there's some people on the low end here enjoy near complete protection from heart disease, from heart attack. Like none of them have had heart attack in this lowest percentile of score. But on the upper end, there are 10, 11% of that bin have had heart attack. There's a more than 20-fold gradient across strata of risk of coronary artery disease based on something measurable at birth, this set of polymorphisms. Okay? Another way to look at this is going back to the distribution here and then ask quantitatively, if you label a group at the tail, the one tail, the right tail, the high risk tail here in red, as carrier for a disease, carrier for a genetic factor, and you label everybody else as non-carrier, then you can ask, what's the risk involved? What's the risk for these carriers? And the reason to do this kind of analysis is actually to bring it on the same terms, an apples-to-apples comparison with the Mendelian mutation analysis. For Mendelian mutations, remember, the analysis is carrier, you have the mutation, and non-carriers, and you're comparing the risk of heart attack in carriers versus non-carriers. And as I mentioned to you, for LDL receptor mutations, 1 in 200 carry the mutation, 199 out of 200 don't carry the mutation, and if you carry the mutation, you're fourfold higher risk for MI. So we wondered, if we label the top 5% of this distribution as carriers, what would their risk be for MI compared to everybody else? And that risk was 3.3-fold. If you go out to a top 1% versus the remaining 99, it was more like 5-fold. So what we're seeing now for the first time, that based on a polygenic model, we can find a strata of the population that is at equivalent risk, basically, to Mendelian mutations. Okay? And this was not really possible before a couple of years ago for maybe reasons we can get into during the question and answer session. But pretty exciting, I think, for the first time we can find a subset at quite high risk based on this polygenic model. Now, all that data that I showed you is actually from one study in the UK Biobank, this study of 500,000 people from England. We wanted to look at the same score that we've developed now in the United States. And we turned to a study of people who've been hospitalized in the United States with heart attack at a young age. This is now 2,000 early onset MI patients from this Virgo study and 3,800 controls from this other study, population-based study. And across all of these individuals, we actually had 30x whole genome sequences generated through the Genomics platform. And so we have then the monogenic model that we can quantify and the polygenic model based on the common variants. And we can ask, of the 2,000 people, how many can we explain potentially using each of these models? So what are the contributions of the monogenic and polygenic models to early MI? And I'm going to just present these data in terms of in a pictorial format. If you take 100 patients with early MI, what we're finding is about 2, for about 2%, it's the monogenic model. And they're anywhere from 3 to 4-fold increased risk for MI based on carrying the mutation in any of those 4 genes that I mentioned. Now, these monogenic individuals typically have a blood test that's also abnormal beyond the genetics. So here's a look at the LDL cholesterol level in people who carry the monogenic mutations on the right and those who don't here. And what you can see is that the LDL cholesterol is quite a bit higher in the carriers with the mutation versus non-carriers. And that makes sense, right? Because the mutations, the monogenic mutations typically work through either elevating LDL or elevating triglycerides, okay? Now, that's the monogenic model. What about the polygenic model in this set of 2,000 individuals with early MI? Turns out nearly 17 of them are high polygenic risk as defined by the top 5% of the distribution. And they're also at similar risk, 3 to 4-fold increased risk. So a much larger fraction of the population gets to early MI based on the polygenic model. And then the other point to make is these high polygenic risk individuals cannot be distinguished right now based on blood tests that we use in clinical practice, like LDL cholesterol. So here's the blood test results for LDL in the high polygenic risk versus everybody else. You can see it's a little higher, but not remarkably higher, right? So to summarize then, polygenic score identifies more than 10x more individuals than monogenic mutations at risk. And this is the prevalence of the genetic risk of monogenic and polygenic in early MI patients, 1.7% versus 17%. They're at similar levels of risk. You can find these monogenic individuals either based on genotype, sequencing and finding the mutation, or the LDL cholesterol. These individuals, I mentioned, they're currently unaware of their risk. The only way to find them is looking at their genotypes. And then thinking about the mechanism of risk, we know here it's related to ApoB-containing lipoproteins. Here, it truly is, for lack of a better term, a gemisch of thousands of mechanisms, or hundreds, at least hundreds of pathways. And so you can't really point to a single reason, single pathway that is leading to for the polygenic model. And then an important clinical question is, what can you do about it? If you find these people as high risk, either the monogenic or polygenic folks, can you actually help them modify their risk in some way? We know that lifestyle and also medications to lower the LDL cholesterol work in the monogenic folks. We did not know what would happen to the polygenic folks. Is that risk modifiable? And we've done work over the last couple of years to show that the polygenic risk itself is modifiable, that DNA is not destiny here, that basically you can modify the risk by either an optimal lifestyle, which can cut the risk almost in half, or taking medications like statins to lower LDL cholesterol to cut the risk as well. Now, the statin part of it may be a little counterintuitive to you, because I just spent 10 minutes saying that the polygenic risk folks don't have high LDL cholesterol, but cholesterol lowering actually works in those individuals. And the reason for that is we probably all have too high LDL. The population average of 130 is probably not the ideal physiologic level. The ideal physiologic level is probably more like 60 or 70. So that's why LDL lowering will work in these individuals as well as the people that have extraordinarily high LDL. Okay, so to summarize this portion then, a genome-wide polygenic score is basically a number that captures one's inherited susceptibility to MI based on millions of sites across the genome. And I think in hopefully 5 years, if not 5 to 10 years, most will know this number like we know the LDL cholesterol right now, because this captures something orthogonal to the current factors, and it's modifiable based on lifestyle and or statin therapy. Now, this is work for one disease in developing a polygenic score. As you know, we here and also others around the world have done similar common variant association studies for a range of diseases. So does this approach work for all these other diseases? And the answer is yes. Here's the polygenic risk score for other diseases, including atrial fibrillation, diabetes, inflammatory bowel disease, breast cancer. And here's a way to quantify the tail of the distribution. If you're in this tail, how much higher risk are you? Or actually, what proportion of the population is in the tail that is more than threefold increased risk? And you can see for all of these diseases, there's a group of women and men who, based on their germline DNA, and the score that can be calculated for each of these diseases, they're at extraordinarily high risk. So you can actually foresee, I think in the near future, based on just a simple genotyping chip, you don't need sequencing. This is just chip data plus imputation, like what you get from 23andMe or other genotyping chips, which are pretty inexpensive now, somewhere between $50 and $100, that with that data in hand, you can systematically calculate, for lack of a better term, a report card of risks for a range of diseases and essentially where you fall in the percentile distribution for each of these diseases. And here's an example of 4, and heart attack being the 5th. One of the other conditions that we looked at, I think the finding that I'm maybe most excited about is obesity, actually. Is that we think about weight as largely a lifestyle problem. We yell at people to basically eat better, walk more, to lose weight, and stigmatize people in terms of their weight because it's felt to be largely lifestyle. But we know that obesity weight is highly heritable, suggesting that some are much more susceptible to a obesogenic environment than others. And we wondered whether we could quantify that susceptibility based on a polygenic score for weight. And that's what we did. We calculated, very similar to the MI work, we calculated a score for weight, and it has a very normal distribution. And then what we did was divided that bell curve, into 3 groups: the top 10% of the distribution, the bottom 10%, and the middle. And what I'm going to show you is data from a longitudinal cohort study called the Framingham study, where people were enrolled at age 30 and then followed for an average of 26 years. And they had weight every 4 years from the time of enrollment. So this is 1971 to the mid-90s. And we know that who was basically obese at baseline and how many developed over time. And the metric we looked at was severe obesity, which is BMI over the age of over 40. This weight over body surface area over the age of over 40. And look at this. Here are the event rates for severe obesity according to the obesity polygenic score. On the x-axis is the years of follow-up. On the y-axis is the incidence of severe obesity. And the dark blue is the top decile, the top 10% of polygenic scores. Here's the middle group, and here's the bottom 10. And what you can see is that if you happen to be in the top 10% of the polygenic score, you're at extraordinarily high risk for severe obesity, almost 18% over the 26 years of follow-up. If you're in the bottom of the distribution, you enjoyed near complete protection from severe obesity. Nobody in this group, this is 10% of the population, nobody developed severe obesity. So I find this quite striking and really suggests that obesity, a lot of it is actually your underlying genetic makeup, and that what the environment is doing is essentially unmasking a genetic predisposition. And we all know people, for example, I think, who are in this group, the bottom, who are rail thin and can eat whatever they want, like Brian Daniels there, and I think that they may end up falling into this group here. And imagine if you knew early in life where your child stood. Are they in the 99th percentile here? Would put them at very high risk. Or in this bottom group, you might act differently or intervene differently. So that's kind of where we're headed, I think, in terms of these polygenic scores. Okay, so that's monogenic, polygenic, somatic mutations is the next topic. Here, these are mutations that are not hereditary, that you're not inheriting from your parents, but ones you're acquiring in the course of your life. These are somatic mutations. And we were able to recently show that somatic mutations actually play a role in cardiovascular disease. Usually we think about somatic mutations playing a role in cancer, but here is a new concept in terms of cardiovascular disease. And what this little picture is a micrograph of a bone marrow stem cell, and it says scientists have learned that an accumulation of mutated stem cells in the bone marrow can increase a person's risk of dying from stroke or heart attack. Okay, so this story here starts with this process called clonal hematopoiesis. So we all have stem cells in the bone marrow, hematopoietic stem cells, that give rise to essentially all the blood cells. Imagine a column here of the stem cells. One of them develops a mutation, shown here in green. As a result of getting the somatic mutation that confers a growth advantage, that clone with that mutation grows over time. The clone can develop additional mutations as well. And this process of the clonal expansion after a mutation is called clonal hematopoiesis of indeterminate potential. CHIP is the other acronym. So if you look at the rate of clonal hematopoiesis, this is detectable, by the way, in the blood from exome sequences. So you can just take the standard blood exomes that are done for any reason, analyze that data using the cancer somatic mutation detection pipelines, and quantify the clonal hematopoiesis process. And here's a plot developed by Sid Jaswal. Age on the x-axis, on the y-axis is the frequency of clonal hematopoiesis. And you can see it's an age-dependent process. As by the time you're in your 70s or 80s, almost 10% of the population, 10% of individuals will have a dominant clone in their blood. Okay? So these clones, it turns out, are not, in terms of the genes that are affected, the mutations affect not just a random set of genes in the genome, but actually are concentrated in just several. DNMT3A, TET2, ASXL1, TP53, and JAK2. These are all cancer genes, leukemia genes. And these top 5 make up the majority of genes mutated for clonal hematopoiesis. So not surprisingly, the presence of a clone was associated with a markedly high risk for blood cancer, developing blood cancer over time. That was not a surprise. But Sid also noted that these individuals were at high risk for heart attack as well. That was a bit of a surprise. We didn't really expect that. So we wanted to replicate this observation. So we did work looking at clonal hematopoiesis in patients with early heart attack. And this is work led by Sid, Pradeep Natarajan, who was in the lab at the time, and Ben Ebert, who was a PI in the cancer program here, and Dana-Farber. So here is the data in terms of looking at clonal hematopoiesis in patients with early heart attack. We didn't expect actually anybody who's young, in their 40s or 50s, the people we studied, to have any clonal hematopoiesis because it's an age-dependent process. But surprisingly, we found that we could find some people who had clonal hematopoiesis at a young age, and they were much more likely to have early heart attack, about 4-fold increased odds. And we actually found that about 2 to 3% of early MI cases actually carry clonal mutations, compared to a much, much smaller fraction in the control population. So we now have a phenomenon, clonal hematopoiesis, that seems to increase risk for early MI. But this doesn't quite establish a causal relevance for this process, mainly because this is determined in blood. It's not germline, it's acquired mutation. So this could be just like a fancy marker of aging, for example. So the causal relevance has been established mainly in model systems. And this is, again, work from Sid, where he wondered whether if you could manipulate the TET2 gene in mice, in a mouse model of atherosclerosis, would you exacerbate atherosclerosis? And shown here are experiments where either wild-type mice or mice that are deleted for the LDL receptor, they're atherosclerosis prone. If you give these two groups of mice bone marrow from mice that are deficient in TET2, would you exacerbate atherosclerosis? And what you can see is that if you give bone marrow from TET2 deficient mice, there's actually more atherosclerosis in both the wild type and the knockout animals. So, and this has been replicated by another group, suggesting that TET2 deficiency does exacerbate atherosclerosis, at least in a mouse model. Now, what's the mechanism here? That's also been looked at. And it looks like it's because loss of TET2 leads to heightened inflammation, particularly in a pathway, this IL-1 beta pathway, this NLRP3 inflammasome pathway. Here's data looking at IL-1 beta levels in the mice, in the atherosclerotic plaque of the mice that were given wild-type bone marrow or knockout bone marrow, TET2 knockout bone marrow, and you can see that there's much more IL-1 beta staining in this atherosclerotic plaque. So this raises some interesting possibilities because there's now a monoclonal antibody that's actually available that's targeting IL-1 beta and was proven to reduce disease risk in preventing recurrent heart attack. And so there's now a hypothesis that if you could basically target this antibody to people who carry clonal hematopoiesis, that the benefit here may be even greater, and that hypothesis is being tested. Okay, so this is a summary then of genetic basis of heart attack, monogenic model, polygenic model, somatic mutations, and the fraction of population with early MI that is affected by each of these models, and then the fold increase in risk. Okay. So let me move the last couple of minutes to this concept of protection. I'll just give you one story to highlight the idea. This is now thinking about genome to identify pathways protective for disease. So if you have a group of individuals who are at average risk for disease, you're looking at their genome, they're at average risk for disease, you might have another group where they carry a mutation, but instead of being at increased risk for disease because of the mutation, they're actually at lower risk, they're protected. So why might you want to know about this phenomenon? Well, it's not really about the individuals who carry the mutation because they're protected, so they don't really need to intervene in them per se. But the reason to know about these pathways, these mutations, these protective mutations, is that if we could develop a medicine that mimics that mutation, then we may be able to transfer the benefit that these people were naturally endowed to the rest of the population and do some good. So that's the concept. Identify these protected mutations, develop medicines that mimic the mutations to reduce disease risk. And it's actually helpful in terms of a specific type of mutation in terms of the protective effect. And specifically, you're looking for null mutations, mutations that completely break a gene, either nonsense, splice site, or frame shift, that protect because then you get a clear direction of effect in terms of which direction you want to manipulate the gene to lower disease risk. And since most pharmacologic interventions actually inhibit a gene's function, finding null mutations that reduce disease risk is actually particularly valuable. So in addition to finding people who may lack one copy of the gene and are protected from disease, which might give you insight into the efficacy of intervention, it's also helpful to find people who might lack both copies of a gene, so-called human knockouts. And if they're healthy, then that might give you insight into safety of completely inhibiting a protein's function. So these are the concepts. Heterozygous null mutation, reduced disease risk, gives you insight into efficacy of potentially mimicking that mutation, and then homozygous null gives you insight into safety. The poster child for this concept is a gene called PCSK9, where 1 in 50 African Americans in the United States carries a null mutation in the gene. As a result of carrying the mutation, they have lifelong lower LDL cholesterol, and they're markedly protected from risk of heart attack, 80% lower risk. This suggested that if you could develop a medicine that mimicked the mutation, that would have similar properties. In addition, it's been few people have been identified that completely lack the gene. They're homozygous null, and they're healthy. So suggesting that complete inhibition might be safe. And so as a result, several companies have developed monoclonal antibodies that block circulating PCSK9. These medicines are now clinically approved for use in the United States. They've been shown to lower LDL cholesterol, and in a couple of trials, they've been shown to lower risk of heart attack as anticipated by the human genetics. Okay, so are there more protective pathways beyond LDL cholesterol, beyond low LDL? The pathway that we've worked on is triglyceride-rich lipoproteins. And specifically, I'm going to give you a story about a gene called apolipoprotein C3. And this story starts with this group. These are the Lancaster Amish. They're a few hundred people from Switzerland in the early 1700s, emigrated from Switzerland to the United States. They stayed a closed population for the last few hundred years, expanded to more than 100,000 people. And so they're really a founder population in the US. They live in the Lancaster, Pennsylvania area. Several years ago, Tony Poland from the University of Maryland showed that 1 in 20 of the Amish carried a mutation that broke one copy of the circulating, of the ApoC3 gene, apolipoprotein C3 gene. And we showed that 1 in 150 in the United States actually carried the same kind of mutation that broke one copy of ApoC3. As a result of carrying these mutations, individuals have lifelong lower triglyceride levels, and they enjoy protection from heart attack. Now, so that suggests that therapeutic interventions that might block ApoC3 would be efficacious in terms of heart disease. But what about the homozygous null? Could we find people who completely lack ApoC3 and give us insight into, again, safety of complete inhibition? So we looked at about 200,000 people in the United States, didn't find a single person who was homozygous null. Then we turned to an important concept in human genetics called consanguinity. That's when parents are closely related to each other, like let's say first cousins, their children are much more likely to be homozygous at any given segment of the genome. And here, consanguinity rates from around the world, darker red is higher rate. And you can see there's a lot of first cousin marriages in northern Africa, in the Middle East, and some parts of India. And the highest rate in the world of consanguinity is actually in Pakistan. More than 50% of marriages are first cousins. And we had a collaborator, Danish Salahin, who was worked with us for many years to ascertain individuals there for a study of cardiometabolic disease. And we worked with him to sequence, exome sequence, tens of thousands of individuals from this cohort. Here's data from the first 7,000. On the x-axis is the genotype of these individuals at the apolipoprotein C3 gene. And remarkably, we were able to find 4 individuals who were complete knockouts for ApoC3 that had no circulating apolipoprotein C3. This is a protein that's measurable in the blood. On the x-axis, on the y-axis here is the blood ApoC3 level. And this is the gene that's involved in your body's ability to clear dietary fat. So after the couple of pieces of pizza that you just had, usually the blood fat level rises. And this gene normally blocks the body's ability to clear dietary fat. So it's a bad actor. The level of C3 is measurable, as I said. We found people who are homozygous null. And here's one of the 4 probands here. We brought this nuclear family back for additional investigation, specifically in terms of how they handle dietary fat. And this dashed line, the 2 lines here indicate that these 2 individuals are first cousins. So we have the father is human knockout for ApoC3, and we were shocked to learn that the mother was also a human knockout for ApoC3, making all 9 children, which is very good for human genetics, homozygous null obligate. So we went from finding nobody who's a knockout to 11 just in this family. So In this family is larger, it's more extended, and in this family you can find all three genotypes: homozygous null, heterozygous null, and wild type. I don't like using the word wild type when referring to humans, but it is that way here in terms of carrying both working alleles. And what we did was ended up bringing back in the same family people who didn't have any mutation and those who were homozygous null. And then to understand how they handle dietary fat, we gave each of the two groups basically 80 grams of fat, big fat milkshake. And this is all work done by Danish Salehin in Pakistan by his collaborators. And we measured blood every hour for 6 hours after the meal in these two groups of individuals. So here's the data. On the x-axis is time, on the y-axis is the blood triglyceride levels. This is unfortunately what's happening to each of us when we eat a fatty meal. In the subsequent 6 hours, the blood triglycerides rises by 2 to 3-fold. Okay? Here's what's happening in the people who are homozygous null for apolipoprotein C3. There's basically no postprandial rise in triglycerides. So now really potentially giving a mechanism as to why these people are protected from heart attack. Literally, these people's arteries are seeing less fat on a daily basis because their blood triglycerides don't budge after a meal. Okay, so these data suggest that it would be helpful probably to inhibit this protein and probably would be safe. And that's what a couple of companies are trying to do. This is work from Ionis, where there's an antisense oligonucleotide that's been developed that decreases the synthesis of ApoC3 in the liver. This has been given to humans. It lowers blood triglycerides, and studies are being planned to test whether it lowers MI risk as well. So I wanted to share with you the story of this concept of protected mutations, developing medicines that mimic the mutations. Now, it turns out for cardiovascular disease, there's now a large number of these that have been identified. There's 7 total protected mutation stories. The PCSK9 was the first. 5 out of the 7 were reported from our group. This is from UT Southwestern, and then ASGR1 is from Decode. So these are all the rates of protected mutations, the blood level that's changed, and then the level of risk reduction, and then where the medicine's development is for each of these. Now, it turns out several of these genes fall into one pathway, and that's this pathway of the body clearing dietary fat. The way lipoprotein lipase, the key enzyme, clears dietary fat in terms of through lipolysis. So this seems to be a key protective pathway for MI. Okay, what I try to show you then is that it's possible now to identify people at risk for MI based on either a monogenic model, a polygenic model, or somatic mutations, and then to intervene in them to reduce disease risk. It's also possible to identify protected mutations and use those as a guide for new medicines development and the identification of new pathways. Thank you.
We might have time for one question. Before anybody have anything they'd like to ask?
Your interpretation of the polygenic risk score, how it fits in, and you said that, well, that may just be that LDLs at baseline are already too high in humans in current standard. Could it also be that basically the polygenic risk score is just amplifying the downstream response? They're hypersensitive to the LDL itself. I was curious more about that second alternative.
Yeah, I mean that's a good point. Yes, it could be that this set of individuals have essentially accelerated atherosclerosis for a given level of LDL, and I think that is also at play. What's in general for coronary artery disease, there's basically a handful of causal paths, and they all seem to just stack on top of each other. They're additive. So one causal path is LDL. Another would be the triglyceride-rich lipoproteins. Another would be like blood pressure. Another might be inflammation. Another is going to be this polygenic background. And so they all seem to kind of add up, or they're all additive, basically.
Thank you again.
Thank you very much.